Hello students, in today's class we will be studying about the biogeochemical cycling of one of the primary nutrients that is required for the survival of all living organisms, that is nitrogen. Now nitrogen as an element is the most common constituent of plants after carbon and oxygen. Whenever we study about any cycle, we need to know about the reservoir of that particular element, the ecological significance or the biological significance of that element, and then the cycle with its various microbial transformations. When we talk about the reservoir of nitrogen, it is present mainly in the atmosphere in the form of dinitrogen or nitrogen gas. In the soil, it is seen in the form of ammonia or even as inorganic ions such as nitrite, nitrate, and we will be studying about them in the next few slides. In the organic form, nitrogen can be seen in living as well as dead matter. So in living beings, whether it is microorganism, plant, animal, human beings, in living beings it is found in the form of proteins, amino acids, and all the living constituents have this, or all the living biomolecules have this particular element. Even in the dead matter, which is present in the soil or in the water sediments, the nitrogen is present in the organic form until it is decomposed. So when we talk about reservoir, mainly there are three reservoirs of nitrogen, that is the atmosphere in the form of dinitrogen gas, in the soil in the form of ammonia and inorganic ions, and in the organic form in living as well as dead matter. When we come to the ecological significance of nitrogen, like I just told you, it is found, or it is an important part of a critical part of amino acids, which means it is a critical part of all the proteins that are found in the bodies of different living beings. It is found in the nucleic acid, in the form, it is found in plants in the chlorophyll molecule. So all the important biomolecules contain nitrogen as their core component or core element. Now before we get into the cycle as such, I would like to introduce you to two terminologies. One is mineralization. Mineralization of any element is the conversion of that element from the organic form into the inorganic form. Mineralization is always the conversion of an element from its organic to inorganic form. And immobilization is the conversion of an element from the inorganic form to the organic form. An easy way for you to remember is I for immobilization, I for inorganic to organic. So immobilization is inorganic to organic form, and mineralization is the opposite, that is organic to inorganic form. Now let us get into the cycle. This is the nitrogen cycle in which every process has been denoted in red. For example, denitrification, nitrogen fixation, Anammox, assimilation, ammonification, etc. Generally, the upper part of the cycle shows the processes carried out under oxic or aerobic conditions, and the lower half of the cycle shows the processes carried out under anoxic or anaerobic conditions. Please take a moment to note down the cycle so that it will be easier for you when I explain the microorganisms involved in each step in the next slide.
Now coming to the first step of nitrogen cycle, that is nitrogen fixation. Now nitrogen fixation, as you can see over here, is a process that can be carried out under aerobic as well as under anaerobic conditions. So under aerobic conditions, we have a few organisms that carry out nitrogen fixation. This includes free living as well as symbiotic organisms. The free living aerobic organisms are Azotobacter, Bigeringia, and symbiotic include Rhizobium, Bradyrhizobium, Frankia, etc. There are many more. We will be dealing about nitrogen fixation in detail in the next few classes. We have anaerobic organisms as well. Those include Clostridium, Chlorobium, Chromatium, Rhodomicrobium, etc. Now, what is this nitrogen fixation? Nitrogen fixation is the conversion of unusable dinitrogen, which is present in the atmosphere, into a usable form, that is ammonia. Now, the nitrogen which is present in the atmosphere constitutes for almost 79% of the Earth's atmosphere, yet it is unusable because here the two nitrogen atoms are bound by triple bond, which requires very high energy to break. So, those organisms which have been listed over here are containing an enzyme called as nitrogenase that helps to break down this dinitrogen into ammonia. This kind of nitrogen fixation, which is carried out by microorganisms, is called as biological nitrogen fixation. We also have other processes such as lightning, which have enough energy to break the triple bond of nitrogen and fix the nitrogen into ammonia. But that constitutes a very small percentage of nitrogen fixation. Maximum nitrogen fixation seen in the environment is through BNF, which stands for biological nitrogen fixation. Now, this biological nitrogen fixation converts dinitrogen, which was an unusable form, into this ammonia, which is present in the soil. The next step is conversion of this ammonia, which is present in the soil, into nitrite and then further into nitrate. This process is called as nitrification. The microbial oxidation of ammonia into nitrite and nitrate is called as nitrification, which is a two-step process. In the first step, we have ammonia oxidation, that is, ammonia is being converted into nitrite. And in the next step, we have nitrite oxidation, where nitrite is being converted into nitrate. So, this two step process is called as nitrification. Now, nitrification can be carried out by chemoautotrophic organisms as well as by heterotrophic organisms. Chemoautotrophs are dominant mainly in neutral and alkaline soils, and they are basically bacteria. Also, these bacteria carry out the nitrification in a much faster process in comparison to heterotrophs. These nitrifying bacteria or these chemoautotrophic nitrifying bacteria are gram negative, and they can use either ammonia or nitrite as an energy source or even as an electron source. So the organisms which carry out ammonia oxidation, chemoautotrophic or bacteria which carry out the ammonia oxidation include Nitrosomonas, Nitrosobibrio, Nitrosospira, Nitrosococcus, Nitrosolobus, etc. Those chemoautotrophs which carry out the nitrite oxidation include Nitrococcus, Nitrobacter, and Nitrospira. These are the chemoautotrophic nitrifying bacteria. Like I told you, we also have heterotrophic nitrifying organisms. These are organisms which are found in the acidic soils and can include bacteria as well as fungi. The process is slower than that of the chemoautotrophic nitrification, and examples of the organisms include Arthrobacter, Aerobacter, Mycobacterium, Streptomyces, Pseudomonas, Aspergillus, and Penicillium. So these are the organisms which carry out nitrification, which is an aerobic process. It constitutes of two steps, ammonia oxidation and nitrite oxidation, and can be carried out by chemoautotrophs or by heterotrophs. This is about nitrification process. The next process is assimilation process. Now, assimilation can be also called as immobilization, which is the conversion of inorganic to organic form. This assimilation or immobilization is shown by all the plants and by microorganisms. There can be two types of assimilation. Either we can have ammonia assimilation, which is shown over here. So we can have ammonia assimilation to form the NH2 amino groups of proteins. So we can have this process aerobically as well as anaerobically. Or there can be nitrate assimilation, which is also called as assimilatory nitrate reduction. So first, let us talk about ammonia assimilation. Like I told you, assimilation is also called as immobilization, wherein this ammonia, which is an inorganic form, is being converted or is being incorporated, assimilated into an organic form, which is basically in the form of amino acids of that particular organism. Now, in ammonia assimilation, the assimilation process can happen either by glutamate dehydrogenase pathway, which is seen when the ammonia concentration is more, or it can be seen by the GS-GOGAT pathway. GS-GOGAT pathway is stands for glutamine synthetase, glutamate synthetase pathway, glutamate synthase pathway. This GS-GOGAT pathway or glutamate dehydrogenase pathway are capable of assimilating ammonia which is present in the soil into an organic form, that is the amino groups of a particular amino acid or a protein. So this process can be shown by all plants and microorganisms that are surviving in the soil. So the ammonia in the soil is now incorporated into that particular living organism. We also have nitrate assimilation. Now, nitrate assimilation is also called as assimilatory nitrate reduction. This nitrate assimilation is carried out by assimilatory nitrate reductase enzyme, and it is mainly carried out by the GS-GOGAT pathway. This again can be carried out by all microorganisms or plants. So plants and microorganisms growing in the soil can incorporate the nitrogen into their own bodies, either by taking it in the form of ammonia or by taking it in the form of nitrate. This process is called as assimilation or immobilization. The next process is dissimilatory nitrate reduction. Now, dissimilatory nitrate reduction is also where nitrate is being reduced into different forms. But here again, we have two processes happening. Dissimilatory nitrate reduction to ammonium, which is abbreviated as DNRA. Dissimilatory nitrate reduction to ammonium is a purely anaerobic process which is carried out by organisms such as Begatoa, Thioploca, Aspergillus terreus, and Chevanella. In this process, we have conversion of nitrate into nitrite by the enzyme nitrate reductase, and nitrite is being converted into ammonia by the enzyme nitrite reductase. So we have two enzymes here, nitrate reductase and nitrite reductase, which are converting nitrate into ammonia. This is an anaerobic process, and here basically we have reduction of nitrate into ammonia. This is called as dissimilatory nitrate reduction to ammonium. The other dissimilatory nitrate reduction is denitrification. Denitrification is carried out by facultative anaerobes like Pseudomonas, alkali genes, Paracoccus denitrificans, Agrobacterium, Thiobacillus denitrificans, Bradyrhizobium, Blastobacter. There are many organisms which carry out denitrification. Denitrification is a type of dissimilatory nitrate reduction wherein we have conversion of nitrate into nitrite. In between, we have two more intermediates, that is nitric oxide, nitrous oxide, to finally give us dinitrogen or nitrogen. So here it is a process of complete conversion of nitrate into dinitrogen. This process is called as denitrification. Denitrification is also a type of dissimilatory nitrate reduction. Why do we call it as dissimilatory nitrate reduction? Because here the nitrate is being used as a terminal electron acceptor, or it is being reduced not for assimilating into the body, not for converting into amino acid, but just as a terminal electron acceptor for energy production. So dissimilatory nitrate reduction can be of two types. One is conversion of nitrate into ammonia, that is DNRA, and one is conversion of nitrate into nitrogen, that is denitrification. Both are examples of dissimilatory nitrate reduction. The next process that we have is ammonification. Ammonification is a process that can be carried out both aerobically as well as anaerobically. I am marking it over here. So ammonification is, in other words, mineralization. Like I told you, mineralization is where organic form is being converted into inorganic form. So when organisms die, it could be plants, animals, microorganisms, whenever any organism dies, it gets degraded, and the organic matter in it, that is the proteins, amino acids, etc., are being completely degraded. They are being completely broken down into to release inorganic nitrogen in the form of ammonium. So we have organisms such as Pseudomonas, Proteus, Serratia, Alternaria, Clostridium, which are carrying out this process either aerobically or anaerobically to release ammonia. So whatever amino group is present in the form of proteins or in the form of amino acid is now being degraded to release back or to put back the ammonium into the soil. This process is called as ammonification, which can happen either aerobically or anaerobically. If it is an aerobic process, along with ammonia, we also get, or the byproducts are carbon dioxide, sulfates, and water. But if it is an anaerobic process, along with ammonia, there there is release of hydrogen sulfide and mercaptan. So only the byproducts are different, but the main product is always ammonia in the process of ammonification. And if the soil is rich in carbohydrate wastes or organic waste, then ammonification is less, because always the microorganism will prefer to first break down the carbohydrate in comparison to the nitrogen waste. But the nitrogen waste is always degraded to release back ammonia into the soil. Coming to the last process, that is Anammox. This is a relatively newer process that was discovered in the nitrogen cycle. Anammox stands for, or it is an acronym for, anaerobic ammonia oxidation. Anaerobic ammonia oxidation is also called as Anammox. So initially it was thought that ammonia oxidation can happen only aerobically. Ammonia can be oxidized only aerobically into nitrate. But then it was discovered that there are organisms which can convert ammonia, or which can oxidize ammonia under anoxic conditions to give back the same dinitrogen. So here we had ammonia oxidation. Ammonia was being oxidized to form nitrite and nitrate under aerobic conditions, whereas under anaerobic conditions, ammonia gets oxidized into dinitrogen. So ammonia combines with nitrite to form dinitrogen. This process is called as Anammox, or anaerobic ammonia oxidation. Now, this is carried out by organisms such as Brocadia or Brocadia anamoxidans, Quinina, Anammoxoglobus, Jetinia, Scalindua. Amongst all of these, the Jetinia, Brocadia, Quinina, and Anammoxoglobus are freshwater species, whereas Scalindua is a marine water organism. But all of these organisms are known to carry out the process of Anammox, that is anaerobic ammonia oxidation, where ammonia along with nitrite gets converted into dinitrogen. This is a relatively recent process. So these 6 steps are involved in the nitrogen cycle: nitrogen fixation, nitrification, assimilation, dissimilatory nitrate reduction, ammonification, and Anammox. This is a tabular column to just show you the names of different organisms that are carrying out each one of the processes that we have just discussed. So nitrogen fixation, like I told you, can occur both aerobically as well as anaerobically. It is carried out by both free living as well as symbiotic microorganisms. Then we have nitrification. Nitrification is a process that is carried out only aerobically, wherein there is conversion of ammonia into nitrite and then nitrate. This is carried out by chemoautotrophs as well as heterotrophs. Chemoautotrophs are more common and they are contributing for more percentage of nitrification because it is a faster process. Then we have assimilation. Assimilation is carried out by all the plants and microorganisms because it is basically uptake of nitrate or uptake of ammonia from the soil and incorporating it into their own amino acids or proteins. This is shown by all the plants and microorganisms living in the soil. Then we have ammonification. Ammonification is a process that is carried out by organisms both aerobically and anaerobically, where they are degrading the organisms or degrading the organic matter and converting it into inorganic ammonium. So here we have conversion of organic into inorganic matter. Then we have Anammox process. Anammox is anaerobic ammonia oxidation, wherein there is conversion of ammonia in combination with nitrite to release dinitrogen. This is an anaerobic process that is seen by a few organisms such as Brocadia anamoxidans, Quinina, Anammoxoglobus, Jetinia, and Scalindua. Finally, we have dissimilatory nitrate reduction. Under dissimilatory nitrate reduction, we have 2 processes, that is dissimilatory nitrate reduction to ammonium, which is an anaerobic process, and denitrification. Though it is an anaerobic process, it can be also carried out by facultative anaerobes. So dissimilatory nitrate reduction is carried out by organisms such as Beggiatua, Thioploca, Shevanella, etc., which convert nitrate into ammonium. And denitrification is a process where nitrate is being reduced to nitrogen. So we have nitrate being converted to nitrite, nitrite being converted into nitric oxide, then nitrous oxide, and finally dinitrogen. This is the process of denitrification. So these are the different steps of nitrogen cycle. I hope you all have understood. Thank you.